You'll see some overlap here. We've discussed muscle physiology in depth, and obviously cardiac tissue behaves similar in a lot of ways to muscle tissue, so you'll see this again. But what you will notice, one of the big differences is, the only part really in all of this that may be a little hard to grasp and wrap your head around is back to the actual physiology of an impulse, right? We talked about chapter 4, and sodium influx, potassium efflux, all that kind of stuff. Well, this is all coming back, but actually it's sort of the same principle here, but you'll see that different ions now. Calcium plays a bigger role, sodium plays a lesser role, and the little sequence of events is a little bit different. So you got to learn the same thing again, but with a little bit different twist to it. Okay, so you'll see what I mean when we get there. But real quick, I want to make sure that we're on the same page in terms of basic anatomy of the heart and the path of the blood through the heart. Okay, so if it's been a little while since you had this, let's make sure we're up to speed. Blood comes in, gathers from the body, and comes to the superior and the inferior vena cava, right? And that brings in blood to the right side of the heart. Let me rewind.
Could you please repeat that?
No.
You said blood comes in.
All right. The blood comes into the right side. Now, when it comes in, it empties into the right atrium. You're going to have to know this anatomy because I'm going to be referring back to it a little bit later. We know the right atrium is where the process begins. CO2 blood enters the heart. You know that song? No? Nobody?
What song is it?
No Potsy's? No Happy Days fans? How we learn the path of blood through the heart? CO2, the right atrium is where the process begins. The CO2 blood enters the heart to the right atrium, the bicuspid valve, the tricuspid valve, the pulmonary arteries and lungs. Once inside the lungs, dumps carbon dioxide, picks up its oxygen supply, then it's back through the heart through pulmonary veins, atrium, and left ventricle. I don't know. Anyways, there was a little kazoo part in there. Yeah, St. John's had it on a commercial. That's right. Yeah. Anyways, when the atria hit, realize that when they contract, both of them contract simultaneously, the right and the left atrium. Okay, not the right side and the left side, but the top and the bottom contract together. So let's look at it from one side. As the blood comes in through here, it receives an impulse, it contracts, it squirts it through this valve. What is this valve? That's right, it's not listed up here, but it's also called the tricuspid valve. You see 3 anchors there, right? These are the chordae tendineae. These are the papillary muscles. Good job. And the way this is designed is to just sort of like be a loose flap that whenever it hits, it pressurizes it to force it through here, so that whenever this relaxes and this builds up pressure, it sort of floats back and shoves this shut. Okay? All right, so once it's through here into the right ventricle, the right ventricle contracts, and it closes this valve, pushes it up through the semilunar valve. In this case, we got the pulmonary semilunar valve. Okay? And out through the pulmonary artery, and this is the only place in the body, of course, where arteries are carrying deoxygenated blood, right? Most of the time they're carrying oxygenated blood. They're going to go to the lungs. They're going to go to the capillaries and the alveolar sacs in the lungs to undergo gaseous exchange. They're going to pick that back up. You're going to come back in through the pulmonary veins from both lungs. This is the only time veins then are going to be carrying oxygenated blood. Comes into the left atrium. The left atrium contracts, pushes the blood through the bicuspid or the mitral valve. Okay, goes into the left atrium. Left atrium hits, goes out through the aortic semilunar valve, out through the aorta to the rest of the body. Okay, what you're going to see is we're going to, of course, focus heavily on just the heart itself and how it works, how the cells inside of all this work. What's different about what's right in here? This connective tissue that separates the atrium and the ventricle, it's different. What happens down through this septum and back out into this myocardial tissue, that kind of stuff. How is this thing working? Okay, so we've got endo, myo, and epicardium layers. Okay, endo, of course, means within. So I'm talking about the inside lining. It's very smooth for obvious reasons. It actually is part of that tissue ends up forming some blood vessels and that kind of stuff. It sort of fades in and forms the interior as it comes out of the heart, for example, like the aorta or whatever. As it comes out, that interior lining is the endothelium, the endocardium becomes part of the endothelium of the blood vessels. The myocardium is that meaty part, the muscle part, the middle part of the heart tissue that's going to have a lot of contractibility. You've got interlacing bundles of fibers. Now, we've talked about muscle fibers. We talked about how skeletal muscle fibers are very long and end to end. They're stacked with sarcomeres from end to end, all the way down. They don't interlace, they don't weave, they run parallel. Okay? Smooth muscle was those same sarcomeric units, but they were anchored in various spots, and they were all weaved around and stuck in between each other so that the entire muscle then would contract in on itself. Okay? And that makes sense if you think about where your smooth muscle is found. What you will find here is sort of a mix between the two. You'll find these long end-to-end units, okay? Actually cells in this case. In a case of like a bicep, a muscle cell, the individual fiber stretched all the way from the insertion all the way back up to the origin of that muscle. There is no insertion and origin when you think of a heart muscle. If you have a heart, it's an organ, and it goes all the way around. So what you have to do is have this lump of cells that is completely connected all the way around. And the heart is not a perfect square. It's not a perfect sphere. It's sort of irregularly shaped. I can't say that word. Irregularly shaped. Okay? And as such, you've got to have, you can't just wrap it around in little circles. It's got to go around. It's got to branch around. It's got to take some detours. And by having relative organization, pretty well lined up next to each other, but allowing it to branch off when it needs to, you can get a tissue that's, makes this surrounding all the way around the heart, and you could, when it squeezes, then it can contract the whole entire organ and make a contraction in that manner. The epicardium is the outside, of course, the very external layer. Okay, cardiac muscle fibers are interconnected. Now, see, what you don't have is just this one cell, like I said, that wraps all the way around and meets itself on the back end of the heart. Like if you did that and pulled it apart and had opposite ends, you could have a muscle fiber like up here, like I showed you, from here to here. What you've got to have is this not just one cell that wraps all the way around the heart. That would be too far to go, and it just wouldn't work right. So instead, what you have is individual cells, but they're sort of butted against each other as they branch, and you can see little lines where the cells butt together. Okay, they're called intercalated discs. I say page 309. That was in the old book. I don't know what the page number is in the new book. What's right? 304. It looks like in the new book. I'm going to show you this. It's this picture though. I don't know if this might be one I grabbed myself off the internet. But this is real tissue. If you were looking at cardiac tissue, you can certainly see that it's nowhere near like smooth muscle. I mean, it is pretty much in a line. You can see that. Okay? It's relatively organized, and it's striated. You can see these lines, these striations in it from the myosin filaments. However, you'll also see that it's not really clear on this picture, but you can see that like this one will come off and part of it goes that way, and then goes this way. It can like fork and it can branch off. Okay, what you'll see here, I've circled them on this picture, are little places where the ends of cells are coming together and getting riveted together and held together. Okay, that's what these intercalated discs are, and you can see those. All right, now if you look close, they're actually spot riveted together. There's desmosomes that are holding them and anchoring them together. But at the same time, you're also going to see a lot of gap junctions along those areas. Because what you're going to see is, like I said before, cardiac tissue has the ability to conduct its own nerve impulse. And when it does that, it's got to be able to spread that nerve impulse very, very quickly over the entire heart. So gap junctions become very important in terms of sharing ions and moving ions and propagating impulses and that kind of stuff. Okay? So you can see the actin and the myosin. It's all the same as it was in skeletal muscle. Okay, so we have branching fibers that are arranged end to end. And like I said before, you can generate an action. I'm going to show you how in a minute. You can generate an action potential in a cardiac cell, in some cardiac cells, without the help of the nervous system. It's kind of neat. It's sort of, like I said, it kind of build a momentum kind of thing, and I'll show you how that works. Here's what I want you to make sure you understand, though. These gap junctions that we're talking about here that allow cells to communicate with one another, allow ions to pass freely between them, these do not occur between the atrial and ventricular cells. In other words, that little area, if you go back to your picture and look at 2, 3, 4 slides earlier, whatever, when I show you the picture of the heart, you can see where the atria and the ventricle become separated from one another. It's where those valves are, that same plane that the valves are on. What you will find is the cells that make up that little area that divide the atria and the ventricle, those cells do not share gap junctions with one another. And you should be thankful that they don't, because if they did, what would happen is your heart would not beat into two different places. It wouldn't be a lub-dub. It would be just a constant contraction. The whole heart would beat as one on one beat, and you can see that wouldn't gain anything. That would not move the blood out and bring new blood in. It's got to be a sequence. It's got to be one then the other. So if they were allowed to communicate via gap junctions, you got to realize that when an impulse would have started in the atria, it would immediately have spread throughout the entire heart. This is insulation, is what this is. Think of it like insulation. It keeps that message from being sent from the atria just through the cells themselves straight into the ventricles. We have the ability to pass that on. That's where the whole, remember we talked about, you learned about the SA node and the AV node and the bundle of His, Purkinje fibers and that kind of stuff. I'll bring that back if you don't remember. But that's what's going to carry that nerve impulse through the ventricles, okay? Not just the cells being active in the atria, okay? All right. So there's also, not only is there not gap junctions between these neighboring cells, between the atria and the ventricles, but they are also separated by a non-conductive fibrous skeleton. In other words, some fibrous connective tissue that's very durable, that makes up the valves especially, that is very non-conducive. It doesn't take an action potential, and it sort of dies out and allows it to not propagate through that area. Okay. Because we're going back to syncytia. Remember we talked about syncytia. We said that there was one cell, and as long as they're all connected by gap junctions, whatever happens in one immediately spreads to the rest of them. That's what's going to happen in the heart, and that's why what I just said was so important, so that whenever something happens to an atrial cell, it doesn't happen to all the ventricle cells as well. Okay. Now, we have an all or none contraction in terms of cardiac muscle. Now, with individual muscle fibers, in the skeletal muscle that we talked about, it's all or none. However, the entire muscle, when you have a skeletal muscle like your bicep or whatever, your entire muscle is not an all or none contraction, right? Because it has to do with the number of motor units that you recruit. The stronger the contraction, the strength of the contraction depends upon how many motor units you are recruiting and how many individual fibers are contracting at that point. That's different than what we get with the cardiac tissue, because there are no motor unit feeds for different fibers to do that partial contraction in the heart. There's not like motor units that feed in and a couple of them get stimulated so you get a little bit of a contraction. That's not the way it works. Your cardiac tissue is all or none. It's the same. It's wired to be the same strength all the time when it hits. Now, it doesn't mean it's always going to be, because something will affect it. You can have varying degrees of the hit, of the contraction of the heart, but it's not going to be because of the number of fibers recruited. It's going to be because of calcium, and I'll show you why a little bit later. It has to do with the levels of calcium that are present. Now, cardiac muscle cells have a whole lot of mitochondria for obvious reasons, and a whole bunch of myoglobin. Remember, what does myoglobin do? You remember? What does it look like? What other word? Hemoglobin. What does hemoglobin do? Carries what? Transfers oxygen. Myoglobin, myo means muscle, right? Myoglobin is very important because it's a special thing, sort of like hemoglobin, that's located in muscle cells that facilitate the transport of oxygen throughout muscle tissue. Okay, so it's very important. Okay, another neat thing is no new cardiac muscle cells are produced after infancy. Remember when I said that you don't, like whenever you're lifting weights and stuff, you don't build new muscle fibers. You take the muscle fibers that you have and you thicken them. Same principle applies. Okay, so the actual number of fibers more fibers are not produced after infancy, but the fibers themselves can thicken and can add in the protein content that they contain. Okay, now we are electrical beings, whether we want to consider ourselves such or not. Up here, all we have is a bunch of electrical activity firing that makes us conscious, that makes us have the decisions and the emotions and everything about us. The motions that we make, the steps we take, I could write a song about that, right? The motions that we make, the steps that we take. All of these things all have to do with electrical activity and impulses being sent throughout the body to cause all this happen at a molecular level. Now, in the heart, certainly that is no different. But what you will see is something called autorhythmicity. Okay? There are some cells in the heart that are autorhythmic. They have the ability to, all by themselves, without being stimulated by an external source, repeat these patterns, these rhythms, over and over and over, which is your heartbeat. Okay? Some cardiac autorhythmic cells display pacemaker activity. In other words, they make the pace. They set the pace. They override everything else that's going on and decide how frequent these impulses are going to be sent, and thus how frequent these contractions are going to be. Okay? Now you got, of all of the cardiac cells present that make up heart tissue, there are two types that we're going to discuss. Okay? Two of the big categories are contractile cells and just conducting cells. These what's called pacemaker type cells. Now, realize that the bulk of the heart tissue is made of the actual contractile cells. 99% of the cells that you have present in your heart are going to be the ones that go squeeze. Okay, they're going to take the actin and the myosin, ration them together, and cause a muscle contraction. Some cells that you have in your heart do not do that. They are not participatory in the contraction. Instead, what they do is they initiate the contraction. Okay, they are like nerve cells that are there that are going to initiate this contraction. Okay. Now, as opposed to reading through this, I'm going to let you do that. I'm going to explain this with this picture. Okay? You've seen something similar to this before back when we did neuronal physiology. Okay? Except the difference was back then it was graded potentials. If it reached threshold potential, sodium gates would open. It would come flying in. Potassium would come rushing out. It would overshoot. ATP-driven sodium-potassium pump would put it back to normal, and you're back again until you get another resting potential. Or another action potential, sorry. The resting membrane on one of those normal cells would be what? You remember? -70. Okay, we're different. Okay, we're a little bit different now. Now with the resting potential is going to be more important in contractile cells, and I'll get to that in a minute. But I want you to look, just right now, keep in mind what we're looking at and what I'm talking about. I'm talking about the cells that are in the heart that have the ability to be autorhythmic, okay? Not the contractile cells. I'm talking about cells that are going to generate this action potential and then thus affect everything else in the heart. So that 1% of cells. That's what we're looking at here, because you're going to see another chart for the contractile cells in a minute. They actually behave differently. Okay, what is happening is, it's hard to pick a place to start because there's no real beginning. It's just an endless cycle. But let's say that you know, based upon your previous experience, that when an action potential is over, the way that you can reset it and get back to and repolarize this cell is for potassium to come rushing out, right? Potassium goes rushing out of the cell and it drops this spike down, and then it sits there until threshold is reached again, until another action potential can start. Okay, here's what happens though in these cells that's different. Potassium does leave and it does come rushing out, okay? However, what's different in these autorhythmic cells in the heart is that there are the membrane of these cells becomes less permeable to potassium as it is falling, as this number is falling, as it is becoming more negative in value down here, okay, as it is becoming more polar. What happens is this slows down, okay? And at about right in here somewhere, potassium was flying out. But what ends up happening is the cell starts closing off the channels through which potassium is leaving. So it sort of trickles out, okay, and it slows down. It starts to slow down. Now, when it starts to slow down, what's important here for you to realize is what is happening at the same time. So imagine this in your head. Picture a cell and picture potassium in a mad rush leaving that cell. And then over a short period of time, gradually, it slows down. It's almost like the channels through which it was traveling starts to close off. And now fewer and fewer are allowed to leave, and it sort of slows down, and the traffic slows down of the potassium leaving. At the same time, imagine this. As those potassiums are doing that, as they are exiting the cell, at the same time, sodium is constantly leaking in. We know there's leak channels. What's different about this situation than what we've talked about before is that you see no sodium movement no bulk sodium movement, no voltage-gated sodium channels, they're not going to happen. There are none here in these contractile cells, or in these autorhythmic cells of the heart. What you have is constantly open leaking sodium channels. Okay? Now, which way is sodium constantly going to leak? It's going to leak where? Which way is sodium going to come? Sodium's going to come in. Okay? So imagine what's happening here. As you constantly have sodium leaking in, and you have potassium slowing down its way out, what you get is a gradual climb of positive charge inside the cell, right? Try to imagine this. If you imagine that the cell was too positive and it's shooting out potassiums in order to drop it back down to this negative down here, to bring it back to where it was, if that starts to slow down, and at the same time as this is slowing down, sodium is still rushing in, what you do is you sort of create this backlog, and more positive gets hung back and trapped back inside that cell. And what you get is a slow rise, and that continues to happen until right in here. And when it rises up, just say in this case about 10 millivolts, as it rises up to about right here, we have voltage-gated calcium channels. This is different now. Now this is before this threshold. Okay, it's important to note. So potassium is effluxing; it's leaving the cell, but it starts to at a certain charge right here. Those gates start to change their chemistry, and it slows down how fast that potassium can leave. When it starts to slow down, it starts to get backed up inside the cell. Well, you couple that with the fact that sodium's constantly leaking into the cell, you're gaining positivity inside that cell. So you get this little climb that you see here. Now, as this is climbing, once it reaches about right in here, that's enough of an electrical charge to change and open calcium channels and allow calcium to come in to the cell. Right? Increased potassium comes into the cell. There's two types. This is where it gets tricky. There's two types of voltage-gated calcium channels. There's what's called transient channels, which are fleeting. They're sort of open. They're not very long. They just happen for a moment. And there's longer lasting. Okay? Now, as this potassium is slowing down on its leak, on its efflux, and the sodium is constantly leaking in, this gradually climbs until some transient calcium channels can open. And when those start to open, guess what? Even more positive starts coming in. More calcium can come in and drop this even closer to zero. Okay? Now, once it gets to threshold, longer lasting calcium channels can open. And when they do, that's a huge influx. That's like with the sodium back in our other cells that we've talked about. In this case, you get a mad rush of calcium in. And when you do, the counter to that, again, is like we started over here, potassium comes flying out. When potassium comes flying out, it does so, and then it gradually slows down. And as it slows down, at the same time sodium is constantly leaking in, climbs back up, calcium ion channels open, becomes even more positive, threshold is reached, more longer lasting calcium channels open, comes flying in, you're back to the same thing over and over. So what you will see is rhythms. You start seeing that this does it by itself. What is not shot through here is an impulse from the nervous system. It doesn't require a receptor coming in, using chemical transmitters, docking on there, opening up channels, allowing sodium to come in. That's not what's happening here. These cells keep this inside themselves. They are able to control these cycles, as you can see, back and forth. And the space between them signifies the space between the impulse ascent and thus the space between the muscle contractions of the heart. It does not rely upon the nervous system. Okay? Now, don't get me wrong. The nervous system can override this. If you get scared, if you get whatever, instantly impulses can be sent that override this and will cause it to increase. Take the normal pacemaking and cause it to increase or decrease, depending upon the situation. But normally, baseline, what controls this is just these cells themselves and manipulating these ion channels like you see here. Okay? Yeah, you're letting now even more calcium come rushing in. That's right. And when you do, that's why this was this rapid spike. Now, what I do want you to notice here is this action potential is actually a little bit longer. If you remember back, there's not a time down here. I'll show you some times later, but in regular cells, this action potential is much, much quicker. and this hump is closer together. It shoots up and straight back down. Here you'll see that it's drawn out a little bit more. And that makes sense if you think about the fact that a normal muscle twitch and a heartbeat, the heartbeat lasts longer. You can actually, you can sort of measure that time. It lasts significantly longer, at least 3 times longer, if not sometimes more. Okay? So this whole thing takes a little bit longer, so it allows a longer duration of contraction as well when it does that. And it also keeps it from tetanus. It keeps it from getting stimulated again. I'll explain that a little bit later. Yes, both of these are voltage-gated channels. Yes. Sorry. Both of them, because they get triggered. Anytime they get triggered, you're dealing with the voltage-gated channel. These weren't open. They became open. They got triggered by this slowly creeping up positive charge. Okay. Questions about that? Good. I'll have you write an essay on about that. Location of autorhythmic cells. Where are these guys at? This goes back to some anatomy that you had, labeling the SA node and that kind of thing. Those nodes that you learn, it's always in blue in the book. It's like a little blue dot between the atria, and then it goes down, and then it splits down through the septum and comes back up the muscles of the ventricle. Those little dots and those colors, that blue path, that's actually just a trace, a roadmap of where these cells are that you can find that have this capability to do this. There's the names, the SA node, the AV node, bundle of His, and Purkinje fibers. It looks like, I noticed this when I was looking over this, that I'm a little bit off on where my line is, but I'll show you. So up here in your right atrium is actually your pacemaker. Okay? Your SA node. Now notice that is actually hardwired, so to speak. These cells all propagate next to each other. The collection of cells, all of these cells that you see in blue have the ability to do what I just showed you. They have the ability to what's called drift and fire. They sort of drift, reach potential, fire. Drift, reach potential, fire. Okay? All of these cells in blue have that potential. However, what you will see is that they each have there's a hierarchy. There's someone in charge of them all, and it's this guy over here, your SA node. That's your pacemaker. He or she distributes that charge accordingly, and you can see how it trickles down. There has to be a delay. Realize that by doing this and by setting these cells, the cells here are supposed to be have the fastest period of that cycling, of that drift and fire. The cells down here are supposed to have the slowest version of that drift and fire, to go boom, boom, bump, bump, instead of blah, blah, blah. Okay? So what happens is this will fire first. I'll show you when we dissect an EKG in a little bit later how you can explain all these electrical impulses and the lines that you see on the EKG. But this will hit first. That impulse will travel almost instantly to the left atrium. They hit at the same time. Then it has to travel through the AV node, and that is sort of like I don't know if you don't know about electricity, but sort of like a resistor, or like something that creates a delay or holds up some of that potential and lets go of less of it. It'll delay it right here so that then it will be in charge of disseminating that information and spreading it back out this way. This is important because what you got to realize is, going back to what I said a while ago, right in here, there are no gap junctions between these cells, and this is an insulated tissue, a fibrous tissue here. Because when this sends out, and this can affect a bunch of syncytia when it does this, when it starts to stimulate and it fizzes out to these cells, if this wasn't insulated and separated, all of this would just immediately all spread out, all to the heart at the exact same time. So we got to have this path to control that and to keep it in order. Okay? So we've got the SA node, the AV node here, the left branch of the bundle of His. This is supposed to be the right branch, but the line came over too far, so obviously it's talking about this right here. And those little branching fibers even further are the Purkinje fibers. Now, things can go wrong with this naturally. What this shows is that usually what drives this train is your SA node. Okay, that's the main engine. That's going to determine the rate and the speed at which these contractions and these impulses are being sent. So if that in fact remains the fastest, just like on this train, if that car remains the fastest, that engine remains the fastest, that's how fast that train can travel. Okay, so that's just like your heart rate. It's going to be as fast as the fastest autorhythmic tissue. if something happens to your SA node, in this case, if it's derailed, if something happens to it, what will happen then is the next thing in line that's still connected to everything, whatever's fastest next is going to determine the rate. In this case, your AV node. Okay? What happens when worse things can happen because here there was still some connectivity. If you look, these two are still connected, right? So this would still exert influence on this. If you look at the next picture, this is separated. The AV node is fizzing out. Something's wrong with its ability to become excited and control its excitation, whatever. This is still functioning. That's still functioning. But the problem is they are no longer connected. So as a result, they function independently. So this could really screw you up because your SA node is still going to go, your atria are still going to go normally according to your pacemaker cells. But your ventricles, where these Purkinje fibers spread out to, are going to be much delayed because they're not getting their faster feed coming from the AV node. So things aren't going to line up. They're not going to be in sync. It's going to be irregular. Okay? Sometimes what can happen too is what's called an ectopic focus. Something spazzes out down here, like especially around in the ventricles in the Purkinje fiber region. If something happens, whether it's electrolytes got screwed up, or there's a lot of things that could happen that could cause these autorhythmic cells to screw up and not maintain their normal rhythms. If it spazzes out completely and it starts to shoot a whole bunch of frequent impulses, we call that an ectopic focus, and when it does, it causes irregularities in the ventricles, and the contraction of the ventricles, maybe defibrillation. A lot of bad things could happen as a result of this. So it will be driven, the whole entire heart actually, this will push forward, push everything forward, and drive and end up controlling, in other words, the entire rate of the heart muscle in that case. So, okay, this is a neat little, I want to show you this picture. So, you'll want to go back and look at this, I think. If you go to this website, scroll down to this picture. Oh, this ain't the one, but this one's fine. It shows you, but I have one in a minute. It's going to show you what the EKG weighs and everything. But you can see how it fires. Okay, the message starts with the SA node. It's kind of fast, but you can follow it. See how it simultaneously goes over to the left atrium, and then there's a little bit of a delay before it gets to the ventricles. Okay, and the actual impulse will be delivered, to these cells actually down here towards the bottom before it comes up the side. So actually, it sort of works its way up. It's a natural sort of movement that would work. Okay. There's some other things you can read about on that page about that if you want to. Okay. Well, that's probably broken now. Tough stuff. All right, contractile cells. Okay, we're going a little bit different way now. We're going to look at not the autorhythmic cells, but the ones that receive this message, and then in response to that, contract. Okay. Now, the resting potential is different now. The resting potential of these cardiac contractile cells is not negative 70, it is negative 90. Okay? Negative 90. It was closer to negative 60. I don't know if your book actually gives a value for it or not. But that's not going to be... where we care most about resting potential is cells that are going to receive and initiate a contraction, that kind of stuff. So we're focusing on that here. It actually, just like before, reverses back to a positive 30, okay, and we'll come back down. This is going to be from a sodium influx, like you've seen before. But what's different about this now than anything else that we've talked about is it will hold this whenever you get this spike in this influx and you get this climbing, okay, this depolarization that is happening all the way and climbing up to a positive region. What you will see is instead of dropping straight back down with that potassium efflux, it sort of comes down and then sort of just trickles down, and it slows and sort of plateaus off and then drops down. You get a delay, okay? That is contractile cells. And it makes sense that that's your heartbeat. You can hear it. You can feel it. You can hear it. All that kinds of stuff. It's a longer contraction. Okay? It's sustained. That contraction is sustained longer. It doesn't just happen and just twitch like a muscle contraction, normal muscle contraction would. So you actually, it'll hold that peak. Okay? It'll hold that contraction for hundreds of milliseconds. Now realize that, see, what's a normal muscle twitch? Like maybe 2 milliseconds? Something like that. I'm pretty sure it's about 2 milliseconds, so like 2/1000ths of a second. This is 200,000ths of a second. So I mean, it seems like not much, but when you're dealing with milliseconds and that small of a number, 200 versus 2 is a big difference between the two. Okay, so you'll get this plateau in the action potential, and I'll show you that. Now, what causes this plateau? Because the other one wasn't confusing enough. Now we got to talk about contractile cells. Again, we're going to see a different role of calcium here. Okay, let's take a look. I ought to let you guys teach this. Split it up. Everybody has a unit. See if we can learn anything from it. Some classes that'll work. I'm not sure it'll work in this one. Let you make the test questions. It's like anybody asks, is this be on a test? Nah. It won't be on there. Don't worry about it. All right, now, I want you to notice a couple things that are different about this graph of this potential. Now, again, we're dealing with cardiac contractile cells. They are not autorhythmic, but they do, just like all muscle cells, have the ability to carry an electrical impulse. Okay? So if you look at this looks different than the other ones we've seen, because first of all, we said that this was a negative 90 resting potential. Fine. But this peak, where it shoots up in the process of depolarization, is extremely abrupt and extremely fast, much more so than anything we've looked at so far. Okay? There's not a gradual approach to threshold like in muscle cells, regular muscle cells. There's not a drifting in because of the leaky sodium and the slow-moving potassium like we saw with autorhythmic cells. No, we're talking a straight shot here. I mean, almost vertical climb. Because whenever they receive this impulse, it's got a lot of ground to cover. That SA node and that stuff we've been talking about that's creating this autorhythmicity and sending that signal, it's got to get out to the entire heart tissue, and this all has to happen really fast. It's got to cover a lot of ground. So what you'll see is the second that excitability is started and goes out into the contractile cells, instantly voltage-gated sodium channels open. There's no approach to threshold. There's nothing like that. They just open. And when they open, massive flood, very, very quickly. Huge influx of sodium into those cells. Okay, now what happens when that occurs? What you have here is immediately, just like before, potassium wants to come rushing out. And it starts to. It starts to come rushing out so as to turn this back the other way. But, like before with autorhythmic cells, you will see that the number is different. We're up in a positive region now as opposed to a negative region, but these cells are different. They have different types of gates on their membranes. Okay? On these particular cells, they have gates that as soon as this starts to happen, this opens the potassium gates to allow them to come flying out, those potassium ions to come flying out. But the second they start to fly out, it's almost like they start to close again. Okay? So they open and they close. And as they close, guess what? The potassium starts to slow down. Okay? It's slowing down. Now, at the same time, what is triggered from this electrical difference are calcium ions. Okay? Calcium channels are open. Okay? Actually, let's look at this. I'm going to re-say this because I think I might have just said something wrong. So I'm going to try to clarify without doing too much damage here. Same thing here. Sodium channels open, huge spike. In response, potassium comes rushing out. I said something about potassium channels closing, going slow. Nix that. What happens here is sodium comes rushing in and potassium tries to come rushing out. Okay? And it does. Here's the problem. The reason that it can't just shoot straight down and the reason it gets dragged on is because at this potential, what opens is not the potassium's not the problem. What opens here are calcium channels. And when calcium channels open, it allows them to run in. So realize what's happening. running into the cell creates this line going up. Taking positives out of the cell brings it back down. The role of potassium is to go out of the cell to bring it back down. Well, guess what? If you have calcium coming back in, that's antagonistic to the potassium. They're working against each other. So they fight and they fight and they fight, and it creates this little plateau that you see here until a point happens where just enough charge is reached to where, bam, the calcium channels close. And when they do, now those potassiums can keep rushing out without anybody working against them. Okay? And then it plummets right back down. Now, what is the benefit to this? I'll go over it again. But what is the benefit of this? A 250 millisecond delay is the benefit to this. Because by creating this plateau, you can increase the time associated with this impulse, thus increase the time associated with that contraction. Okay? And I'll show you it does. Yes, it lengthens it lessens the frequency of the impulse, and it lengthens the degree of the duration of the contraction. I'm going to show you another picture in a second, explain why that's really important. But one more time, negative 90 resting. You've got autorhythmic cells doing that drift and fire thing. Okay? As they are doing that, they are sending periodic, very rhythmic impulses throughout the cardiac tissue. Some of the cells are contractile cells, which would behave in this manner. They would receive that impulse. Instantly when they do, their sodium ion gates would open. Sodium would come just blitzing in, go straight up, bring it all the way up to the peak, and in so doing, potassium would want to counter that and come rushing out. So it does. It starts to turn and it starts to leave potassium. But it says slow because it's slow in terms of...
Okay, so potassium's trying to get out.
It is. It's leaving. It's leaving.
It is leaving, but it's leaving.
But we don't... It is because it's sort of like congestion. You've got... Think of it... It's not exactly right, but think of it like that. Potassium's trying to leave, but at the same time, calcium's trying to come in. So as a result, what do you see? You don't see a net change. It's sort of like congestion. Okay? Potassium's coming out. It is leaving. But you do not see this charge drop back down to where it was. Because they're working against each other, calcium is coming in while potassium is going out. So as long as there is this opposition here, in a battle for charge, as long as there's a battle of where the positives are, this is going to drag this on, and it's going to keep dragging on until one of them gives up, basically. In which, if calcium would have given up, if calcium gives up, you get what you see here. Those gates close, calcium can't come anymore, potassium is still allowed to leave, so then it can hurry up and leave. And it's got less resistance, it'll go, it'll leave, and you'll see this plummet. Had the other happened, if they were fighting against each other and the potassium gave up, guess what you would see on this graph? A huge spike upward. It would continue to go even farther because calcium would have continued to come into the cell. Okay? So they're working against each other.
So finally the calcium channels.
Yes. Right around in here. The calcium channels start to close, and when they start to close one by one, it happens very quickly. Boom. All of a sudden, there's not that opposition between potassium and calcium, and now potassium continues to just keep going where it was going, and it plummets down. No, ignore that. Because that's tricky. Because what they're saying there, increased... Yeah, just don't worry about that. It has to do with levels. Increased levels of potassium. Increased movement of potassium, decreased movement of calcium. That's what that's saying. So. All right. Got that? Piece of cake. Okay, so it's complicated stuff. We take it for granted, right? So, summarize. Let's look at what's going on. We've got action potentials in cardiac contractile cells when they receive that. They have received that from the autorhythmic cells. Okay, so if you have these contractile cells that receive this impulse from the autorhythmic cells, when they receive it, remember, their anatomy is very similar to skeletal muscles in the sense those T-tubules, those little invaginations that I talked about, how it dips down in and goes in, and there's sarcoplasmic reticulum next to it, and there's calcium stores, and they get divvied out, and they go down to the actin and myosin, all that kind of stuff. There was a picture of that. You'll need to know how that that works, the general anatomy of that. Well, those same T-tubules will be found in cardiac tissue as well, cardiac contractile cells. So that nerve impulse then will travel through the contractile cells. It'll go into those T-tubules, go down into the guts of that cell, into where the actin and myosin is, and realize that what you don't have in cardiac cells is near the calcium stores in those sarcoplasmic reticulum, those sacs that store a lot of calcium in skeletal muscle. There's not near that much stored calcium in cardiac cells as there is in muscles and in skeletal muscle cells. So most of that has to come just from floating around in the extracellular fluid, is where this calcium becomes very important. That's one of the reasons that a calcium disruption in the diet, calcium and potassium especially, yes, it can affect nervous impulses and muscle contractions, but especially cardiac situations, because the cardiac situation depends upon calcium from the ECF, not just from stores that it can control within it. Okay? All right, so you've got a small amount of calcium from your extracellular fluid that will be triggered, okay, if that impulse travels down those T-tubules. Now, you are going to get a release of what calcium is stored, okay, from those reticulum, sarcoplasmic reticulum. So you get an increased release of calcium floating around now that can come and interact with the actin, basically, the troponin tropomyosin complex, and pull that cord back and expose those binding sites. Okay, so with increased calcium now in that cell, this can happen, right? And then you can get cross-bridge interaction. Then you can get sliding filaments, get a muscle contraction as a result of that. Now, there's a larger pool of calcium, okay? In the cardiac cells, in the actual cytosol, floating around calcium, there's actually a larger pool of calcium to draw from, not stored in sarcoplasmic reticulum necessarily. Just floating around calcium, there's a larger pool in cardiac contractile cells than there is in skeletal muscle cells. Okay? Because, remember that the muscle contraction is dependent upon how fast calcium is pulled back out and tucked away and pulled off of that complex. As long as calcium is bonded to the troponin tropomyosin complex, you're going to continue to get a muscle contraction. If you want that to relax, you've got to pull that calcium back and store it back up to let those fibers relax from one another, or those little filaments come apart. But in the case of cardiac tissue, you'll see there's a much larger pool, so as a result, much less of it gets taken away at any given time, or at least more slowly. And as a result, the contractions last longer, about 3 times as long. And like I say here, the amount of calcium is what's actually going to determine how much cross-bridge activity you get. I said a while ago that it was all or none in cardiac tissue in terms of the individual fibers. Either the heart receives an impulse and it contracts, or it does not. In the case of skeletal muscle, you can recruit different numbers of fibers to increase the contraction of the whole muscle. Here, you're going to control how much contraction happens based upon how much calcium is present. Okay? If there's a little bit of calcium present, you're only going to get a little bit of contraction. You're only going to get a little bit of movement, a little bit of ratcheting movement, because there's a larger pool it has to be dealt with. Okay? So if there's not as much there, you're dependent upon that for the strength of your contraction. The more calcium that's present, the more interaction you're going to get at the cross bridges, okay? And the more contraction that you're going to get. Okay, digitalis is a drug, right, that's used in heart conditions that can actually help increase the strength of muscle contractions, increase the strength of cardiac contraction. How do you think it might work? What do you think it does, based upon what I just said? It's written in the same font as the other 2 things I wrote up here. Probably has to do with what? Calcium. If you want to increase the force of the contraction, what might that drug be able to do? Increase the duration and increase the force of that contraction. What will it do with respect to calcium? Will it pull it out of there faster, or will it make it linger around a little bit longer? Yeah, digitalis makes calcium stay a little bit longer in these areas, in these cytosol areas, and it helps block the reuptake of calcium to help facilitate better cardiac contraction. Okay, what I want you to see here, and it's not really labeled on here, but it is in your book, is these things are superimposed on each other, and they're different. This is the action potential of what goes through a contractile cell, a cardiac contractile cell. This is actually the timing and the duration of the actual contraction of the muscle. Now, what I want to point out is that when you have skeletal muscle, the time, the entire duration of the action potential is actually much, much shorter than the entire duration of the contraction of that skeletal muscle. In other words, you've got this plateau phase, right, in cardiac situations. In normal skeletal muscle situations, this goes up and straight back down. And then you get a contraction that would last, a decent amount of time for that fiber. What I'm saying is, when you have a muscle contraction, you get a fire, and you could actually fire another action potential before the actual fiber contraction was done, before it was over. That's how you get some together. That's how we could get tetanus, basically. If you could keep firing, because the action potentials are so fast in skeletal muscle compared to the length of the actual contraction, you could fire them back to back and fire another potential before that contraction was over, and fire another potential before that contraction was over again, and keep building on that until you get a flat line. You get a tetanus situation where you've overstimulated to the point where you get constant contraction now that can't relax. This is prevented by this in the cardiac. We don't get tetanus in the heart, thankfully. In terms of, when I say tetanus, I don't mean infection. I mean, you don't get a locked up muscle contraction. You can get defibrillation and all kinds of crazy stuff, but it doesn't just lock up on you under normal circumstances. Because, notice, the ability to draw this out, this action potential, by drawing it out, almost the entire length of the actual contraction itself, prevents another one from being shot right behind it to prevent tetanus from happening. Okay? It'll get shot, and then it can't fire another one actually until that contraction is over. That's what this shows. That's important. I don't know if you grasp that or not, but it's very important because it prevents tetanus. It prevents from firing back-to-back impulses before that contraction is over. Okay, EKG. Depends if you're dealing with Greek or Latin, whether you want to use the C or the K. Greek is K, I think Latin was the C or one or the other. I forget. But it's still an electrocardiogram. A record of overall spread of electrical activity through the heart. Now, it's not, when you see this, you've probably seen one before. I mean, you're in the health field, you're nurses or whatever. You know what an EKG, at least roughly, looks like. And you see these spikes from the paper. And these are in reference to differences in electrical charges, okay, that occur. If we know that, like with the nodes that we've talked about, it starts there, something fires, and there's an electrical change, we can trace electricity, and we can follow those electrical changes as they happen. And when we do that, we can create these little graphs that you see. Okay, and then those graphs mean something. Now realize, when you take an EKG, you don't just lock stuff up on the heart. You actually put nodes all the way out here. You can put them on your hands, your wrist, on the rib cage, around the heart, the legs. I mean, if you do a full 12-point lead, I mean, they can go all over the body. So what actually this takes advantage of is the fact that electrical potential, you have a lot of water in you, right? It's got a lot of electrolytes dissolved in it, a lot of sodiums and potassiums, all other ions. You have electrical charge that comes from your heart that actually gets dissipated to all your tissues, because that heart is in contact with other fluid that goes in contact with the rest of your body. So when these changes happen and these electrical charges start, they can actually get transferred through the fluid in your body all the way out to like the surface of your skin and your arms. Now, it's very minor, but with instrumentation you can pick it up. I mean, you don't go walking around and just like harnessing all this electrical power or anything. But anyways, that's what it measures is the difference. For example, if you put a lead here and a lead here, it will measure the difference. That's what voltage is. Voltage is potential difference of electricity between 2 points. So if I measure, I've got electricity going through here, and I measure it this compared to this over here, then I see that there's a difference. There might be what's called a volt or a millivolt difference of electricity between what I measured here and what I measured here. Now, electricity itself, it's difficult to give it a unit, but what you can do is compare two places, how two places compare in terms of strength. And that's what this will do for you. So it's not a measure of a single action potential. So don't get confused. When you see an EKG, don't think that you're looking at a firing of an action potential. You're not. It's a sum of electrical activity that's shot all the way around the body. So who knows how many potentials are involved. It's just electrical difference that you see. Okay, you can look through those pictures yourself. I don't expect you to know them. It's just to show you I'm not making it up. Okay, now take a look at this. This obviously would be an illustration, an artistic rendition. This is not really what you would see. It's much more sloppy than this in real life. But let's just say it was perfect and it looked like this. What does this mean? Now, this is not a substitute. It takes medical school and a lot of training to be able to fully interpret everything possible from an EKG. Okay? I'm not going to try to say that you're going to know everything that you need to know when I get done talking about this, so don't make that mistake as a professional either. But I'll give you the basics as to what these things represent. Now, as far as clinical applications thereof, not my ballpark. Okay, so you guys, that's for you to work on yourself. My experience, just so you know, my experience with this is not with humans. It's with laboratory animals and frogs especially. So I'm not going to be able to help you with your daily clinical applications of this. But I can certainly tell you the theory behind it. Okay. All right. What we have here is what's called the PQRST sequence. Okay. Did you guys talk about this already in 102 a little bit? Okay. So let's make sure we know what we're doing here. We've got area. This hump that you see right here is atrial depolarization. I'll explain it more in detail in a second. But depolarization, in other words, contraction. Okay. This is what's going to be the impulse that's sent by the SA node. This hump that you see is because of an impulse that's being sent by the SA node that's going throughout the atria, and then during this, right during and right after it, in this area right here, you're talking about where the atria are actually contracting. Okay. Now, during this time, that impulse is being sent down through, gets to the AV node. Okay, it's getting through that area. Once it hits the AV node, it starts to dissipate, and it starts to go out through the ventricles, and at which point you get ventricle depolarization and you get ventricular contraction, ventricular systole. Okay, remember systole and diastole. One's contraction, one relaxation. Systole is contraction. Now, what is happening at the same time here that you cannot see because it is masked? It's a hard word to say too, especially with how I slur everything together. Masked. It is covered up. What's behind it is atrial repolarization. The atria are relaxing, but you can't see that because the ventricles are so much bigger. There's so much more electricity associated with them. They hide it. Okay. So in this QRS area right in here is where the ventricles are contracting and the atria are relaxing. Okay. At this point, you've got ventricular repolarization. The ventricles relax right here. Then you've got a delay called the TP interval. The delay during which you're waiting for that next drift and fire to happen, that new impulse to be sent. In which case you start over again with another P wave, which you're back over here again. Okay, so this is called the P wave. This is the PR interval right here. You can keep track of where the message is going if you know that you got SA node, AV node, Purkinje fiber kind of thing, and you keep in mind that we've got atria, ventricles relaxing. Okay. Okay. Now, I want to show you something that you'll want to take a look at again a little bit later when you're studying, because I think it's really good. What you can do is, now you can slow this down fortunately, but this is a cool animation, and I'm going to slow it down for you to show you how it'll help you. But what this does is shows you the heart contracting while the nerve message is being sent in yellow. It shows you where it's going, what part of the heart is contracting when it's receiving that nerve impulse, what part of the EKG corresponds to that, what you're seeing, and the relative pressure inside the atria and the ventricle during that time. So it's actually really cool. It helps you see stuff. Now what you can do is go here and slow it down, hit stop, and when you hit stop, what I suggest is go over here to where it says step, and then go back to the beginning, and try to figure out and step through it. Oops. So like here, notice what's happening. I'm at the very beginning, right before the P wave. And you can see that here's my SA node. It's getting ready to fire. So what I should see is the firing of the SA node and the subsequent contraction of the ventricles as a result of that. So we step through it, go a little slower. You can see where the yellow is spread now. It's part of that P wave. The P wave was shot. When it did, look what happened. That little interval right before that spike is where that contraction actually occurred. See that? where the atria are. You can see that they've become more narrow. They've squeezed in on themselves. Now, what happens now is that message was propagated to the AV node. You can see where it's highlighted now. Look at your EKG. Look where we're at. Okay, what it corresponds to. Keep going. That message gets sent now as it is being sent out down through the bundle of his out to the Purkinje fibers. You can see on the EKG what that corresponds to. It's spreading, and now it's getting ready to contract. There it is. There's your contraction. You can see how that relates to the EKG. Now that next hump that you see is bringing it back to normal and relaxing. So then you can see it all together. And one thing that you can do at the same time, if you want to look at that, is whenever the atria hit, obviously the pressure inside them increases. And whenever the ventricles hit, the pressure inside them decreases. And you can look at that pressure gauge down there to see how that happens. Okay. I don't know. Good question. I've not actually looked. I don't think there are. But I don't know if this site you can link off to and try to find other ones. It looks like arrhythmia treatment. I'm not going to that site. Maybe there's some animations on that. But that would be good if there was. You could really learn a lot from that. I wonder if I got a tattoo of an EKG. Thinking about it. I wonder if you could like put it up here and like turn it and make it like. But I could get actin and myosin filaments. Yeah, and put them on your muscle, and then when they contract, you can actually watch the actin and the myosin get smaller. That's awesome. Oh, there'll be a few people in the world. Sweet tattoo, man. Yeah, I know. Hang on a sec. Oh, yeah, we're in good shape. We'll stop there.